Welcome to another lecture by MedicoMedics, Friedreich's Ataxia. In this lecture, we will present a general introduction to Friedreich's Ataxia. We will talk about normal physiology, the development of pathophysiology, key concepts like GAA triple expansion, iron cluster synthesis, reactive oxygen species, and their role in Friedreich's Ataxia. We will continue with symptomatology, diagnosis, and treatment, talk about current research, and end with a summary. Now, Friedreich's Ataxia, FA, is a rare autosomal recessive neurodegenerative disorder characterized by progressive ataxia, muscle weakness, and systemic complications. It is classified as a rare disease, estimated to affect 1 in every 50,000 worldwide. Now, the cause is a mutation in the FXN gene. This results in reduced levels of the protein Frataxin, which is a mitochondrial protein essential for iron-sulfur cluster formation. Thus, the FXN gene is faulty, leads to reduction of Frataxin, which is critical for energy production in cells. Now, we will talk about this pathophysiology and the steps involved here in greater detail as we move through this presentation. Now, inheritance. Now, Friedreich's ataxia is inherited through autosomal recessive inheritance, which means both parents must pass on a defective copy of the gene. The onset symptoms typically begin between 5 to 15 years of age, though late onset forms do exist. However, in late onset, above the age of 25, is associated with slower progression. Now, let's begin with normal physiology and normal frataxin function. But before we talk about the role of frataxin, let us just briefly review the process from DNA to proteins. Now, DNA is made up of genes. These genes contain instructions to make proteins. And what happens is that DNA is transcribed or copied into messenger RNA. Then we have organelles in our cells called ribosomes that read these messenger RNAs and synthesize a protein by linking amino acids according to the genetic code. So, to say it in a different way, we have a recipe, our DNA. Then we have the chef, our messenger RNA. He interprets this genetic code or this recipe and then starts producing the proteins according to the instructions given here. And it's the same with Friedreich's ataxia. One of these genes in our DNA is the Frataxin gene, and it provides instructions to make Frataxin protein. Now, in the next 2 slides, we will talk about the pathophysiology of Friedreich's ataxia and talk about the genetic mutations that have occurred, leading to a reduced level of messenger RNAs, which in turn leads to a reduced number of Frataxin proteins. So, turning our attention back to the role of Frataxin. So Frataxin is a mitochondrial protein, and Frataxin helps regulate iron balance and supports energy production through the process of oxidative phosphorylation. Now, Frataxin is also essential for biogenesis of iron-sulfur clusters, and these are crucial for electron transport and enzymatic reactions. These clusters are required for oxidative phosphorylation, which is the process that mitochondria used to generate ATP, which is the cell's main energy source. Because these mitochondria are considered the powerhouses of the cell, and they are responsible for generating adenosine triphosphate, or ATP, through oxidative phosphorylation. And ATP serves as the primary energy currency of the cell, and it fuels essential processes such as muscle contraction, nerve signaling, cell repair, etc. And without sufficient ATP, cells cannot maintain normal function, leading to energy deficits and even disease. So frataxin plays a crucial role in the formation of these iron-sulfur clusters. And they are, in turn, essential for enzymes in cellular energy production. These clusters support oxidative phosphorylation, enabling for efficient ATP synthesis. Furthermore, frataxin prevents iron accumulation, reducing free radical formation and mitochondrial damage. So you can already see the importance of the frataxin protein on such a fundamental level of ATP synthesis. Now let's dive deeper into the pathophysiology of Friedreich's ataxia. Now, if you remember from the previous slide, we talked about the process from DNA to messenger RNA, from messenger RNA to proteins. And we said that DNA contains genes, and these genes contain the instructions for the synthesis of specific types of proteins. In the case of frataxin, we have the FXN gene, and it contains instructions for the frataxin protein. Now, the genetic issue in Friedreich's ataxia begins with the concept of GAA triplet repeat expansion. Now, what is a GAA expansion? So, GAA triplet repeat refers to a sequence of 3 nucleotides, guanine, adenine, adenine, and they are repeated multiple times in the first intron of the frataxin gene. And if you see here in this illustration, we have normal and we have FRDA, meaning Friedreich's ataxia. And aside from the outcome and a very specific detail written here as 20 and 700 here, the rest looks the same. Because healthy individuals also have frataxin proteins. However, what is the difference? So in healthy individuals, if there are GAA triplet repeats, they typically range from 5 to about 30 repeats, and it does not interfere with FXN gene expression. But in Friedreich's ataxia, the individuals inherit abnormally expanded GAA repeats, so typically between 70 to 1,700 repeats. And this is in both alleles of the frataxin gene, leading to gene silencing. So how does this expanded GAA repeat cause disease? Now, it's important to know that the expanded sequence does not change the frataxin protein itself, meaning there is no direct missense or nonsense mutation. Instead, it causes structural disruptions in the frataxin gene. So what happens is that we have a formation of heterochromatin, meaning that the expanded GAA repeat disrupts chromatin structure, making the FXN gene more compact and less accessible for transcription. And this in turn leads to histone modifications, further silencing FXN expression. And all of this leads to abnormal RNA DNA loops. And this occurs during transcription. So if you remember the illustration from the previous slide, we go from DNA through the process of transcription to messenger RNA. And due to these loops that occur due to the GAA repeat, and due to this silencing that happens, we will have RNA polymerase stalling. RNA polymerase is what reads the transcription from DNA to RNA. So it will lead to RNA polymerase stalling, therefore a reduction in the amount of functional FXN mRNA production. That was a mouthful. Meaning that the problem is we are not producing enough messenger RNA. And if we don't have messenger RNA, we cannot then translate it into functional proteins. So the problem is not getting from messenger RNA to frataxin protein. Rather, the problem is getting from DNA to messenger RNA. So in summary then, because of the GAA triplet repeat expansion, we get gene silencing. We get disruptions making less messenger RNA. So that in turn then leads to less frataxin proteins being synthesized or formed. And as we've mentioned, frataxin is essential for assembling iron-sulfur clusters, and these are essential and needed for mitochondrial enzymes involved in the production of ATP. So without enough clusters, ATP synthesis gets disrupted. Furthermore, since iron-sulfur cluster formation is impaired, iron isn't properly utilized, which leads to a toxic buildup of iron in the mitochondria. This toxicity, i.e., the overload of iron, binds with oxygen to form something called reactive oxygen species, or ROS. They are also known as free radicals. And these free radicals are unstable molecules that damage proteins, lipids, and mitochondrial DNA, which worsens cell dysfunction. So in summary, the lack of ATP production and oxidative damage lead to progressive loss of cells. So we see neurodegeneration happening, muscle dysfunction happening, and these are key symptoms in FA. Now let's dive a little bit deeper into the issue concerning the dysfunction of iron-sulfur cluster formation. Now we have talked about the central role of a disrupted biogenesis of iron-sulfur clusters in Friedreich's ataxia. So here we have two illustrations, and these two illustrate iron-sulfur cluster biogenesis pathways, specifically in the mitochondria. So the first illustration here on the left, it shows how the mitochondria, which is the cell's powerhouse, builds iron-sulfur clusters, and these act like mini batteries that power important proteins for energy production. Now, the NFS1 protein provides the sulfur, while the ISCU2 help build iron-sulfur clusters. Now, what does frataxin do? Seen here. Frataxin plays an important role in regulating this process and ensuring that the clusters are properly formed. Now, once these clusters have been built, they are then transferred by the ISU2 to GLRX5, seen here. And this helps delivering these clusters to mitochondrial proteins. So all in all, what does it mean? It means that the mitochondria act like a factory producing these iron-sulfur clusters, and these are essential power sources for key proteins that keep cells running smoothly. And frataxin is a critical worker in this factory, helping the whole process to run efficiently. Now, in the second illustration, we don't have the frataxin available. So without frataxin, the cluster factory malfunctions. So what happens here? The NFS1 protein will struggle to produce iron-sulfur clusters, and the ISCU2 will fail to properly transfer these clusters. Therefore, we will have fewer clusters built. And because iron is not being used up properly to form these clusters, iron will build up inside the mitochondria. And this buildup of iron, binding to available oxygen, leads to the development of these reactive oxygen species. And this causes oxidative stress, so damage to the cell. So what does all of it mean? Without iron-sulfur clusters, energy production will be severely reduced, leading to nerve and muscle damage. So frataxin is like a missing worker in a factory. Without it, the entire iron-sulfur cluster production line collapses, and there is iron overload and oxidative stress damaging the mitochondria, starving cells of energy, and leading those symptoms seen in, for example, Friedreich's ataxia. Now, before we move on to symptomatology, diagnosis, and treatment of FA, let us try and summarize everything said thus far. So we mentioned that we have a GAA repeat expansion. So on a molecular level, we have abnormally long GAA repeats in intron 1 on the FXN gene. I might add that this is occurring on chromosome 9. Now, because of this, we will have FXN gene silencing. And the clinical outcome is low frataxin levels. Furthermore, these GAA triple repeat expansions cause chromatin to become tightly packed. We call it heterochromatin formation. So we will have compact chromatin that will prevent RNA polymerase from efficiently transcribing the FXN gene, leading to RNA polymerase stalling. So the main issue here is that the expanded GAA repeats make the chromatin structure too tight, blocking the machinery needed to read the gene. So we will have reduced FXN transcription and decreased frataxin mRNA. And if we have decreased frataxin mRNA, we will have decreased frataxin proteins. And once we arrive at a frataxin deficiency, we will have a loss of iron-sulfur cluster assembly. And this in turn will lead to mitochondrial dysfunction and clinical outcomes like neuropathy and muscle weakness. Furthermore, due to the loss of the ability to assemble iron-sulfur clusters, we will have a buildup of iron in the mitochondria. And as iron will bind with available oxygen-derived species, we will have the production and increase of reactive oxygen species. And these increase oxidative stress. And this stress will lead to neuronal and cardiac damage. Clinical outcomes will be ataxia or cardiomyopathy, for example. Now let's revise and repeat the symptomatology in Friedreich's ataxia. So all FA patients will present with neurological features. There will be a loss of large sensory neurons, so proprioception, loss of balance, coordination, loss of reflexes. We will see a loss of spinocerebellar tracts, so a loss of balance and coordination, a loss of dentate nucleus of the cerebellum, so we will see dysarthria, potentially loss of vision due to a loss of the optic nerve. Now, because it is a systemic disease or a multi-system disorder, we can classify them into the following. So in neurological symptoms, we will see ataxia. And if you want a more comprehensive understanding of what ataxia is, please do look at our lecture on what is ataxia. We will have dysarthria, so slurred speech due to impaired muscle coordination, loss of sensation, muscle weakness. We will have musculoskeletal symptoms like scoliosis, seen illustrated here, where we have a curvature of the spine, and this often will require bracing or surgery. Foot deformities, so high arched feet is common. Another crucial part of FA are the cardiac symptoms. And we expect to find hypertrophic cardiomyopathy, so a thickening of the heart muscle, and this in turn leads to arrhythmias or heart failure. So what happens is that, as you can see in this illustration right here, in hypertrophic cardiomyopathy, there is a thickening of the heart muscle, and this is very common in FRDA or Friedreich's ataxia, and it impairs the heart's ability to pump blood effectively. Because let's connect it to FA. So what is happening? In the pathophysiology, when we have frataxin deficiency, as we've mentioned, we would have mitochondrial dysfunction and the loss of energy or energy deficits. We will have iron overload, which causes oxidative stress, damaging these cells. And in this case, it's damaging our cardiac cells, heart cells. Now, when these heart cells are damaged, there will be processes of inflammation and reparation. But the problem is, this in turn, this reparation process will lead to the buildup of fibrosis or scarring, leading to the thickening of these walls. And that in turn reduces the flexibility and pumping efficiency of the heart. So just like any other muscle you are trying to grow, you go and lift weights. What happens is that we create these microscopic tears in our muscle fibers, right? And we let our body repair this by fusing fibers together, leading to thicker and stronger muscles. And this is called muscle hypertrophy. Now, when we go to the gym, That's our intended goal, to get our bicep to grow, let's say. However, when we are talking about the heart, we're talking about a very special structure, and we only have a certain area or space to work with. So to maintain the efficiency of the heart walls, the muscles in the heart cannot overgrow. If they do, the ability to pump will go down. If you think of it like if you are in a room and if you have space to move, you can jump and extend your body fully to then contract. But imagine that room getting smaller and smaller, or inversely, you getting bigger and bigger. Eventually, you cannot stretch your whole body, so you can't then contract properly. So that's essentially what's happening in hypertrophic cardiomyopathy. So what is essentially a good thing, inflammation, reparation, it's happening in a very bad place. And because these heart cells are getting damaged and dying, the reparation is causing this secondary issue, which is the hypertrophy, and that in of itself becomes a pathological issue that can then lead to arrhythmias, for example, or even to heart failure. And this is, of course, also correlated to the other early signs of cardiac involvement, like palpitations and shortness of breath. Other symptoms, like diabetes mellitus, occurs in about 10 to 20% of patients due to pancreatic dysfunction. And then we have, of course, fatigue seen in more or less all these patients, caused by mitochondrial energy deficits. And if you are wondering why diabetes, for example, such a different pathology, why is that happening in patients with FA? Because we have in our pancreas, we have these beta cells that produce insulin. And if you've heard about insulin, you probably know the role it plays in regulating our blood glucose levels or our blood sugar levels. The hormone insulin here is crucial. Now, just like these cardiac cells are getting damaged, so too do these beta cells get damaged. So insulin secretion or production will go down, and regulating glucose levels will become more problematic, eventually enough to become diabetes. Now here we have a table connecting the different symptoms to diagnostic methods and then to whatever available treatment we have. Now, we don't have a cure for Friedreich's ataxia. And what we do is to try and correct for and manage the different symptoms that are present. A severe one is the cardiomyopathy. And we do that, we diagnose it with echocardiography and EKG, and we manage it with the use of beta blockers, ACE inhibitors, and of course cardiac monitoring. But what you see here are a lot of neurological symptoms. So neurological exam will be most prevalent here. And so, for example, if we have progressive gait ataxia, we can provide mobility aids and physical therapy. In dysarthria, so slurred speech, we can provide speech therapy. For muscle weakness or issues in proprioception, physical therapy, strength exercises. In scoliosis, we assess it with x-rays and MRIs, and we can use orthopedic bracers or surgery if it's severe. For diabetes mellitus, if confirmed, we can provide insulin if needed or dietary control. Now, there isn't any necessary list of priorities because this is very individual. You can't say that all patients will have the identical list of symptoms and severities. It will differ. Some will struggle more on the neurological side. Some will have more prevalent issues with their cardiomyopathy. So it's on an individual basis. But I could add that cardiomyopathy, so the most common cause of death in FA is issues with the heart, because hypertrophic cardiomyopathy and heart failure due to this progressive myocardial fibrosis that's happening. These are the leading causes of mortality. So to try and identify that as early as possible and get the correct treatment available. Luckily, thanks to the available treatment we have to cardiac pathology, like the use of beta blockers in dealing with hypertrophy in general, or ACE inhibitors to deal with these are usually medications used in blood pressure management, but they have similar effects here, helping us sustain cardiac effect for as long as possible. Same with diabetes mellitus. Having unregulated blood glucose levels can be fatal, but because we have insulin, we have ways to treat diabetes. We can manage these if they are identified early. Now, let's take a look at what is happening in today's research field. So we have some drugs and new therapies that are both available and up and coming. Now, this is not an extensive list, and there are other drugs in development. But for whoever is watching this right now, in 2025, we have the first FDA approved and available medication or medicine, which is omaveloxolone. What it does is that it activates the NRF2 pathway, which regulates the antioxidant response and mitochondrial function. And what is its connection to FA pathology? Well, in FA pathology, as we've talked about, we have mitochondrial dysfunction and excessive oxidative stress due to low frataxin levels. Now, by activating the NRF2, we enhance the cell's ability to combat oxidative damage and therefore improve mitochondrial energy production. So the expected effect is to reduce oxidative stress, improve energy production, and help slow disease progression. So it's a way to deal with one of the consequences of this inability to produce these iron clusters, because we will have iron overload leading to oxidative stress. So here we try to find a way to reduce that. So it's symptom treatment, but because it's stopping or aiming to stop oxidative stress, and we know the consequences of this oxidative stress and the damage it causes, it could be an option to manage FA. Another one is vatiquinone, which inhibits 1,5-lipooxygenase, an enzyme involved in oxidative stress and inflammation. So again, it's sort of the same logic here. In FA, oxidative stress, again, contributes to nerve cell degeneration and muscle dysfunction. And by blocking this specific enzyme, the idea is to reduce oxidative damage. And this may then protect neurons reduce nerve damage, and potentially preserve motor function over time. We have CTI 1601. Here we have a different mechanism of action where we are delivering frataxin protein directly to the mitochondria using a cell-penetrating peptide technology. Now, this is approaching the problem from a more fundamental level because the primary deficit or defect in Friedreich's ataxia, as we've talked about, is the deficiency of the protein frataxin. Because of the GAA repeats, we couldn't get enough mRNA from this DNA. And because of that, we will not have enough translation into functional protein. And we need this frataxin for iron-sulfur cluster formation in our mitochondria. So without it, the function deteriorates, leading to neurodegeneration and muscle weakness. So it's quite straightforward. It's to increase levels of frataxin, potentially then restoring mitochondrial function and slowing or stopping disease progression. Another interesting idea or area of research is gene therapy approaches. Here the idea is to use viral vectors like the AAV to deliver functional copies of the frataxin gene, or delivering gene editing tools like CRISPR to correct frataxin gene functions. So now we've moved to the fundamental level because this is a genetic disorder. And we talked about the GAA repeats being the cause of all of this, the expansion here, leading to gene silencing, leading to reduced transcription, and in turn then leading to reduced frataxin proteins. So if we can correct this gene deficit, likely all these next steps should follow and we should have an increase of frataxin. Now this would then be a cure, or at least significantly halt the progression in FA. So you can see here that luckily, right now, in the research field, all levels are being approached. On the level of symptoms, to halt, to stop progression. We have the idea to increase frataxin because we've identified that it's low and that the issues that follow with mitochondrial dysfunction are due to the absence of frataxin. So we have an idea here to increase the amount of proteins available. And even on a more fundamental level, we are approaching the gene issue itself to try and correct for all of these. So in summary then, Friedreich's ataxia is a genetic disorder caused by FXN mutations, leading to progressive ataxia, muscle weakness, and systemic complications. Frataxin deficiency disrupts mitochondrial function, causing oxidative stress and damage to nerves, muscles, and the heart. Symptoms include ataxia, scoliosis, hypertrophic cardiomyopathy, and diabetes. We diagnose it via genetic testing, and it's managed with therapies, medications, and emerging approaches in gene therapy, for example. Complications, loss of mobility, heart failure, and diabetes are key challenges. And that's the end of this lecture. Please remember to share, like, and subscribe for more weekly videos.